Hi everybody. Welcome to Chapter 15, Genes and Proteins. This chapter is a continuation of and an expansion upon topics that we have previously discussed in earlier units of the class. We learned previously that a gene is a segment of DNA containing a set of instructions for a particular process or product. We also learned that a trait is defined as an inherited characteristic of an organism, either physical or behavioral. But what we haven't talked about yet is how exactly it is the case that genes give way to traits. What is the actual physical mechanism by which the information in the DNA is read by a living organism cells and then put into action so that it manifests as a trait? So that's what this chapter is going to be all about. And we'll start it off by reviewing just a few of the basics that we should already know, but just in case, we're going to go over them again. Genes are, of course, composed of DNA, which is a type of nucleic acid, and nucleic acids are made of smaller building blocks called nucleotides. Segments of nucleotides are organized into genes, and the order of the letters in the gene, the A's, T's, G's, and C's, spells out the instructions for how the cell should make specifically a protein or RNA molecule. Different versions of the same gene can have different sequences of letters, and these are referred to as alleles, different versions of the same gene. So to paint a little bit clearer of a picture of the significance of DNA being turned into a trait, let's talk about a specific example, which is actually one that we've referred to earlier in the class. We learned before that on human chromosome number 4, there is a gene, and that gene is called the SNCA gene, located at the 4th chromosome on the larger arm at position 22.1. And this section of DNA at this particular locus encodes instructions for making a protein called alpha-synuclein. You may remember that alpha-synuclein is a protein that is implicated in Parkinson's disease, and therefore the trait that it encodes is something to do with nervous system function. So I'm going to show you what the actual sequence of letters is in this piece of DNA, this gene, which writes the instructions for making this nervous system protein. I'm going to go to a database called the National Center for Biotechnology Information, which contains essentially the sum total knowledge of all gene sequences that humans have identified in the human genome as well as in other living organisms. And this database is going to show us a series of letters which represent one of the DNA strands that is involved in this gene. DNA, of course, is a double-stranded molecule, but this database only shows you one strand for convenience sake because knowing the rules of complementary base pairing, you can just assume what the sequence of the second strand is without having it actually there. So we're going to look at this one strand of this gene. Okay, so here we are. We have Homo sapiens gene on chromosome number 4. And down here we have a series of A's, T's, G's, and C's in a particular order, and this is what spells out the instructions for making this protein. So if I scroll down, then you can get a sense of just how big this piece of DNA is. And it keeps going. I'll stop there because there's still more to go, but you get the idea. It's a very long piece of DNA, lots of instructions for making this complex protein that is involved in nervous system function. What all those instructions amount to is a recipe for making this protein that you see on the screen right here, which again is called alpha-synuclein. It's made of 140 amino acids in length. And this protein, we know, is involved in transmitting nervous system impulses from neuron to neuron in the brain. So that's what the instructions encode for, this physical protein right here. Now I'm going to focus on just one segment of those letters, or those instructions. Alpha-synuclein has a very long set of instructions, as we saw, but This screen right here only shows us approximately the first 1,000 letters in those instructions. Now I've highlighted one of those letters right here because this letter has some significance to the disease Parkinson's disease. In the sequence for a normal, good, healthy recipe for making alpha-synuclein, this letter right here at this position, which is the 209th letter, that letter is normally a G. In some people, a mutation can occur, which makes it, instead of a G or a guanine, an A or an adenine. And that might seem like a pretty minor error in the instructions for how long the instructions are, but it turns out that that little error can put a person at risk for Parkinson's disease because it changes the way that the cell reads these instructions, and it changes the way that the protein is made. If the instructions are altered in that specific way, then the protein will be made with a different structure, and that structure actually is so different that it leads to the accumulation of these plaques inside of neurons called Lewy bodies. And those Lewy bodies gum up the inside of the neurons and interfere with nervous system transmissions, which is what leads to the disease Parkinson's disease. So this is just one example of a protein that we've mentioned before and a trait or a disease that it's involved in. So what we are overall getting at in this chapter is we are going to talk about how the cells actually take that sequence of letters that you just saw, read it, and then use that information to synthesize a protein at the end of it all. How do they do that? That's the question that we're answering here. So we're going to start at the very beginning by talking about the DNA. And so you guys know, the version of this process that we're going to be going over in this lecture is the version as it is carried out in eukaryotes, not in prokaryotes. We're going to talk about the more complex version, and that is the version that happens in eukaryotes, which also includes humans, of course. So in eukaryotes, DNA must stay inside of the nucleus. That is where it lives, and that is where it stays. An analogy that we've used before is that chromosomes are like books, and the genes inside of them are like recipes that are written from this four-letter language consisting of A, T, G, and C. It is very, very important for the cell that the books remain inside of the nucleus, which is kind of like the only library that the cell has for all of these recipe books. If there was only one library in the world, it would not be a good idea to let books leave that library, because those books would be the master copies of everything, and so therefore they must be kept safe. If there really was only one library in the world, then the way that people would have to get information from that library is not by checking out the books, but rather by going to the library and making copies of the pages in those books, and then taking those out of the library instead of the originals. And in fact, this is what cells do as well. The cell can only access the information in the genes by making copies of them because it cannot take the DNA out of the nucleus. The copies can be removed from the nucleus because they are temporary. They're not the originals. And so this is the very first step in the mechanism for making the end product, the protein, from the DNA. So for this reason, the process isn't quite as simple as simply reading the DNA and writing the protein. There are actually three separate steps that have to be carried out, the first of which is called transcription. Transcription is where a DNA gene is copied into a molecule of RNA. RNA, you may remember, is that single-stranded molecule, which is like DNA, but it has some differences in its structure. And these copies will be made not in the form of DNA, but rather in the form of RNA. After the RNA copy is made in transcription, then comes post-transcriptional modification. Post-transcriptional modification is where the RNA that is made in transcription is edited and prepared for use in actually making a protein. And finally, the third step is called translation, and this is where the edited RNA sequence is actually used as a blueprint for turning into a protein molecule. So we are going to walk through these 3 steps one by one, and we are going to follow them from the DNA all the way to the end product, which is the protein, and see how this process works. So we'll get started first with transcription. Transcription is the first stage of the overall process, but within transcription there are actually 3 different substages, and those are called initiation, elongation, and termination. They are exactly what they sound like. Initiation means to begin. Elongation refers to the fact that it's going to make this long strand of an RNA copy. And termination refers to the end. So essentially this means to begin, to make the long RNA, and then to end. Before we look at how this works, let's take a look at a gene and its internal parts, because a gene is a little bit more complex than simply a recipe. There are actually different internal parts to a gene. The gene includes, in addition to the recipe, which is shown here in this lime green color, a promoter, which comes at the beginning of the gene, and a terminator, which comes at the end. The promoter is essentially a piece of DNA that comes before the gene, before the recipe, which says the recipe is about to start. The terminator is a little piece of DNA that comes after the gene, which says the recipe is over now. So we can kind of think about this as being like the front cover and the back cover of the book, which signify to you that things are beginning and things are ending, and everything in between is the recipe book. Now that we have a sense of what the gene looks like, we can talk about how transcription works on that gene. In the first stage of transcription, which is called initiation, a special enzyme called RNA polymerase binds to the template RNA at the promoter. Now, RNA polymerase, if this sounds familiar, it's because we previously talked about another similar enzyme called DNA polymerase, specifically DNA polymerase 3 and DNA polymerase 1 when we were discussing DNA replication. DNA polymerases, as we saw, were enzymes that made strands of DNA. So it makes sense then that RNA polymerase is an enzyme that makes strands of RNA. And that's what we're trying to do here in transcription. We're trying to take the DNA and make a copy in the form of RNA. So RNA polymerase will attach itself to the promoter, which remember is the little piece of DNA at the beginning which says the gene is about to start. And RNA polymerase is able to identify the promoter because the promoter always has these specific little sequences of letters in it which indicate that it is indeed a promoter. So RNA polymerase will attach to that thing, and then it will unwind the 2 DNA strands, which allows the sequence of the nitrogenous bases to be exposed. This exact same thing happened when we talked about DNA replication, except the enzyme that was doing the unwinding in order to expose the nitrogenous base sequence was called helicase instead of RNA polymerase. But in this process, helicase is not needed. RNA polymerase is sort of a one-stop shop enzyme that does it all in the process of transcription. It attaches, it opens the DNA, and then when we move into the second stage of transcription, which is called elongation, RNA polymerase will start to cruise through that DNA molecule, opening it up, reading the sequence of letters here, and then building an RNA strand that has a sequence of letters which complements the sequence in the template strand. You may notice that RNA polymerase is only copying one side of the DNA, and that's because, remember, RNA is a single-stranded molecule. We don't want to make multiple strands of RNA. We only want to make a single strand, and so the RNA polymerase only copies what is called the template strand. The RNA strand that is synthesized from this process is called the messenger RNA, which is also abbreviated to mRNA. And it's called the messenger RNA because it is carrying a message, the copy of a message that is encoded in the DNA molecule. Now at this point, let's take a step back and do a review checkpoint. In a double-stranded molecule of DNA, what are the pairs of nitrogenous bases that are always found across from each other on complementary strands? Or in other words, what letters go with what letters between the two strands? So when elongation takes place, as we're going to see in this figure right here, the RNA polymerase follows the same rules for matching up complementary nitrogenous bases, or the letters, as DNA to DNA would, with one exception. RNA does not contain the letter T, or the nitrogenous base thymine, and it instead contains the nitrogenous base U, which stands for uracil. And that's something we learned about previously. So anywhere that a T should appear in the RNA sequence, instead, the RNA polymerase will lay down the letter U. So this is what the building of the messenger RNA sequence looks like. This will go on until RNA polymerase runs into that little sequence at the end of the gene called the terminator. And once RNA polymerase encounters the terminator, this sends a signal to it that the gene has ended, and therefore it releases the DNA and the new messenger RNA, and the new messenger RNA is free to move on to the next step. The next step is called post-transcriptional modification, and as we said before, this is when the RNA sequence has some edits made to it so that it can be prepared for use outside of the nucleus. Of course, transcription has taken place inside of the nucleus because that's the library and the DNA has to stay there, so the copy has to be made in the nucleus. And before that copy can leave the library, it has to have some changes made to it. And those changes altogether amount to post-transcriptional modification. So this is what our messenger RNA looks like after it has been transcribed. It is called the primary transcript, or the pre-mRNA, when it is in this form, because it has not yet been edited or had the changes that are necessary made to it. So pre-mRNA is the name for the RNA that comes out of transcription. After it undergoes post-transcriptional modification, then it's just considered mRNA. During post-transcriptional modification, three distinct things happen to the pre-mRNA. The first thing is that a molecule called a methyl G cap gets attached to the 5' end of the mRNA. The 5' end, of course, is the end that corresponds to a phosphate group. So a special molecular cap gets added to that end, and the purpose of the cap is to stabilize the pre-mRNA molecule and allow it to be recognized by the enzymes that are responsible for the next step, which is translation. The second thing that happens is that a long string of adenine nucleotides, called the poly-A tail, literally meaning the tail of many A's, is added to the opposite end, which is the 3' end of the pre-mRNA, the end that corresponds to the sugar in the backbone. This tail, kind of like the cap, stabilizes the RNA and protects it from being degraded. The third thing that happens during post-transcriptional modification is that entire chunks of the primary transcript are cut out and the remaining chunks are sewn back together. The segments that are chopped out are called introns, and the segments that are sewn back together are called exons. Let's take a look at how this works. So, introns are segments of the messenger RNA that contain special sequences of nucleotides telling the cell that they should be removed. This would be like if you went to the library and made a copy of a recipe for making spaghetti, but there were some parts of that recipe that you didn't like. Maybe you don't like garlic or basil, and so before you left the library, you went and you crossed those things out. That's what's going on here. The cell is actually removing parts of the instructions that it doesn't want in there. So these sequences tell the cell that this is an intron and this needs to be removed. The cell sends out what are called small nuclear ribonucleoproteins, which is abbreviated SNRNPS and is pronounced SNRPs. That is the actual pronunciation of that abbreviation. These SNRPs will bind to these sequences in the intron, and then after they attach themselves to those special intron sequences, they will find each other and come together to form this giant mass called a spliceosome. The spliceosome will cut out the intron and then join the exons together so that it is still a continuous molecule of RNA. The beauty of this process is that it allows for a phenomenon called alternative splicing, which is where different genes, or rather different proteins, can be produced from the same gene depending upon how you cut it. So if this up here is what the pre-mRNA looks like, then depending upon how the cell decides to slice that DNA, it can either come up with one recipe or an alternative recipe that lead to different protein forms. So let's say that this gene at the top, the pre-mRNA, says long, red, white fur. Depending upon whether the cell decides to cut out the white or the red, it can either come up with long red fur or long white fur as its end protein form. And that's the beauty of alternative splicing. It can lead to things like, for example, a snowshoe hare being able to produce white fur during the winter or brown fur during the summer, and therefore being more adequately camouflaged during those times. So what happens to the introns then? Of course, if the introns that are being chopped out don't go on to be part of the final recipe, then they don't contribute to the protein. And for this reason, scientists used to refer to the introns as something called junk DNA, but that term has now been debunked and is considered outdated because scientists now believe that there's no such thing as junk DNA, and in fact the introns go on to play an important role in how genes are turned on or off by different triggers in the environment. In this checkpoint here, what I want you to tell me is what would be the consequence if a pre-mRNA molecule did not have a methyl G cap added during post-transcriptional modification? What would happen? So let's take a moment and take stock of what we have accomplished so far in this process. We've talked about transcription, which is the conversion of the DNA molecule into an RNA copy called a pre-mRNA. And we've also talked about RNA processing, also known as post-transcriptional modification, which takes the pre-mRNA and finalizes it and edits it so that it is then ready to leave the nucleus or the library and go on to the third and final step, which is called translation. During translation, what we are going to see happen is the RNA sequence in that messenger RNA is going to be converted into the final product, which is the protein molecule. So translation takes the info in the mRNA copy and it creates the protein. This all happens inside of the cell's ribosomes, which we learned previously were the structures in the cell responsible for making the proteins, and now we're going to learn how that happens. Ribosomes are giant complexes that are themselves actually made of RNA and protein, and their job is to receive the incoming RNA and turn it into protein. Proteins, as we learned before, are made up of building blocks called amino acids. They are one of the 4 macromolecules, and their monomers that make them up are the amino acids, of which there are 20. Those are the 20 that you see here in this amino acid chart, and remember that they can be divided into groups based upon their chemistry. We have nonpolar amino acids that don't like to jive with water. We have polar amino acids that do like water. And then finally, we have both positively and negatively charged amino acids. So to summarize, the three components that are needed for translation to take place are, of course, the ribosome, which is the protein factory that carries this all out, the messenger RNA, which has the instructions for making the protein, and then finally the amino acids, which are the raw material of the protein. Ribosomes are talented in that they can actually read one language but write another language. They can read RNA, but they can write in protein. The ribosomes do this because they have a special decoding mechanism that allows them to figure out which amino acid should be incorporated into the protein structure based upon the words that it is reading in the RNA message. Specifically, for every 3 RNA nucleotides that the ribosome reads, 1 amino acid is brought into the protein. These sets of 3 nucleotides we can kind of think about as being the words in this cellular language. But instead of being words, they are called codons. So in the example right here, this segment of RNA contains 9 nucleotides, and therefore it would contain 3 three-letter words, which would yield 3 separate amino acids. The amino acids are brought to the ribosome by another special type of molecule that is called a tRNA. tRNA stands for transfer RNA, and is called transfer RNA because its job is to transfer the amino acid, so it's self-explanatory. A transfer RNA is composed of 1 strand of RNA here that has folded on itself several times and hydrogen bonded in several places in order to form this T shape. So what we're looking at is one piece of RNA. It's a single strand of RNA, but it has actually bonded to itself at a few different locations to form this weird wobbly structure here. Every tRNA molecule has a sequence of 3 letters, because remember, this is made of RNA, so it is again made of nucleotides that have the A's, U's, G's, and C's in them. And at one end, the tRNA has a sequence of 3 letters called an anticodon, and these 3 letters end up being important in how the tRNA works. For this particular tRNA molecule, the anticodon reads CGG. But the cell has anticodons and tRNAs for every possible combination of 3 letters down here. This one just specifically happens to be CGG. At the other end of the tRNA molecule, it is carrying an amino acid. And so the way that the tRNA molecule works is it will use this anticodon here to match up with the codon in the messenger RNA, which is why it's called an anticodon. And in doing so, it will deliver its specific amino acid to the ribosome so that the ribosome can use it to build the protein. So this is a picture of how this might work. We've got our tRNA molecule up here. Here we have our anticodon, GAG in this case. This is our messenger RNA coming out of the nucleus, which has sequences of letters that will be divided into sets of 3. So here's the codon, CUC. The anticodon that matches with it is GAG. And the transfer RNA that has this particular anticodon is going to be carrying the amino acid known as leucine up here, one of the 20 different amino acids. So scientists have worked out this code, and so we are actually able to decode and predict what amino acid will be incorporated into the protein depending upon what the codon is in the messenger RNA. And all of this is displayed nicely in these things called codon tables. This codon table shows you every possible set of three-letter words made up of the letters A, U, G, and C, which are the four letters that are found in RNA molecules. Now, the table might look a little bit overwhelming, but there's actually a specific way that you go about reading it, which is that on the left-hand side, the first letter is used to organize the different codons into rows. So if the first letter of the codon is a U, you look in the first row. If it's a C, you look in the second row, etc. The second letter of the codon is listed up at the top. So if the first letter is an A and the second letter is a C, then you would look down here for the first letter being A, and then you would come over to the column where the second letter is a C. And then the third letter is represented on this side here, but usually I find it's easy enough to go based on the first letter and the second letter, and you can find what codon it is you're looking for. Two things that are important to note on here are the fact that there are a few codons that are highlighted in colors here. AUG is highlighted in green because this codon is called the start codon. It is found at the beginning of every gene. It is always the codon that starts things off. The first word is always AUG, and this word corresponds to the amino acid known as methionine, another one of the 20 amino acids. The other one you can see up here are three codons that are highlighted in red, UAA, UAG, and UGA, and these are all referred to as the stop codons, because these codons actually don't encode an amino acid at all. Instead, they tell the ribosome when it should stop translating into amino acids because the protein has ended. A final thing to note about this table is that you can see that there are multiple words that actually encode for the same amino acid. So the table is redundant. You can have two different words, such as, for example, GCU and GCC, both of which encode for the amino acid alanine. So in this checkpoint here, I want you to get a little practice using the codon table by taking the sequence of letters that I have laid out for you here and pretending that this is a messenger RNA molecule that is coming out of the nucleus and ready to be translated. I want you to use the codon table to translate it and tell me the sequence of amino acids that will be made if this is the recipe for the protein. So how you should go about this is you should write the sequence down, you should divide it up into three-letter words, and then you should take those three-letter words and cross-reference them with the codon table, which I have again for you here, to figure out what is the sequence of amino acids that will be incorporated into the protein based on those letters. So now we're going to take a look at how translation is actually accomplished by the ribosome. Ribosomes are made of two distinct pieces that are called subunits. And those two subunits come together and create this sort of space that is the factory for the protein during translation. On the left, we have the small ribosomal subunit, and on the right, we have the large ribosomal subunit. What these two subunits do is they come together and they clasp the messenger RNA transcript, and the mRNA will sort of thread through between the two ribosomal subunits, and it will read the mRNA and translate it into a protein. Biologists consider translation to also have three substages in it, and these substages are referred to with the same names that we use for the three substages of transcription. So translation starts with initiation. Again, to initiate just means to start. Elongation refers to the elongation of the protein molecule that's being made by the ribosome. And then finally, termination means the end of the process, because termination means to end. So here's the messenger RNA molecule that we're going to be working with here. And before we get started, let's note that every messenger RNA molecule will start with a segment of letters that is not actually translated into the protein. It is after this segment of letters, this untranslated region, that we see the start codon, which is always AUG. We can pick that out right here. So as translation gets started, what will happen is the small ribosomal subunit will attach itself to the beginning of the actual protein-making part of the recipe. The small ribosomal subunit enters first, and then before the other part of the ribosome comes in, actually, the first tRNA enters the scene. The first tRNA is called the initiator tRNA, and the initiator tRNA is always carrying the anticodon UAC, which matches up with the start codon AUG. The tRNA is also always carrying the amino acid methionine, which is what the start codon codes for. So the initiator tRNA will bind to the start codon, and then once it binds to the start codon, then the large ribosomal subunit will join the picture and clasp to the mRNA on the other side of the small ribosomal subunit. When it does this, it brings the initiator tRNA into what is called the P site of the ribosome. We can think about the ribosome as having these two rooms in its large subunit. Within those rooms, one of them is called the P site, and the P site is where the growing chain of amino acids is anchored, as we will see. The A site is the other room, which is empty right now, and this is where each new tRNA molecule will enter the ribosome as it starts to read the mRNA. So at this point, we would say that the initiation stage of translation is complete, and the ribosome is now ready to move into the elongation stage, which is where it starts to make this long string of a protein by connecting more amino acids. So the ribosome will read the next sequence of letters in the messenger RNA, which in this case is GGA. A tRNA molecule with the corresponding anticodon sequence, which will be CCU, will approach the A site of the ribosome and enter it. When it does so, the amino acid that is being carried by this new tRNA molecule will form a bond with the amino acid that is currently hanging out in the P site. So now we have two amino acids that are connected to each other. At this point, the ribosome is ready to read the next codon, but of course there's not enough room for another amino acid because the ribosome only has two rooms and they are both occupied. So when this next codon comes along, which in this case is UGU, corresponding to the anticodon ACA, then what's going to happen is the first tRNA molecule, the initiator tRNA, is actually going to get kicked out. It leaves behind its amino acid because that amino acid formed a bond with the amino acid that was previously in the A site. This amino acid is now in the P site because everything has shifted over by one space to read the next codon. Of course, the most recent codon has brought with it a new amino acid. This one is cysteine, abbreviated CYS, and it forms its own bond to the amino acid that is now in the P site, glycine. This process repeats where a new tRNA is called up to match with the next codon, AUGGUC. This one's carrying lysine. And the ribosome moves over to capture that new tRNA, kicking out the tRNA that was in its P site and leaving behind the amino acid that it formed a bond with. The ribosome will repeat this process, moving through the messenger RNA molecule until it encounters one of those three words which we saw were stop codons. At this point, the elongation stage is over and we're moving into the third and final stage of translation, which is called termination. Here we see one of the three stop codons, UAA, and remember that those stop codons don't actually encode for an amino acid. Instead, what happens when a stop codon is encountered by the ribosome is something called a release factor approaches. And the release factor is a molecule that will enter the ribosome, match up with the stop codon, and then send a signal to the ribosome that the protein is over. You have done your job, and therefore you can just disassemble yourself and fall apart. And that's exactly what happens. At this point, translation is complete. So what has it accomplished? Well, we started with a sequence of messenger RNA, what you see at the top, and the cell has now converted that into a sequence of amino acids. We had 15 letters in the messenger RNA, and now down here we have 5 amino acids in this small protein. So now I'd like to show you guys a video that sort of puts together all 3 of these steps: transcription, post-transcriptional modification, and translation, and shows you how they happen as one big seamless event from beginning to end.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
So hopefully that video was helpful for seeing all 3 steps of the process put together. At this point, if you are getting tired and need to take a break, this would be a good time to do it. We're going to move now into another part of the lecture where we talk about a related concept to what we've just been discussing, and that is mutations. So if you need a break, take it now. If not, we are going to press on, and we are going to be asking the question of how does changing the DNA sequence change the protein that is made from that DNA sequence? We are going to look at a few mutations, and we're going to check out what effect they have on the protein. So I've created these hypothetical mutations here where we have an original sequence of a piece of messenger RNA. You can tell that this is the messenger RNA and not the DNA because it has U's instead of T's. And then I have a mutant where I have changed one letter out between the original sequence and the mutant. And we want to find out what effect will this mutation have on the amino acid sequence of the resulting protein. The first step is that you have to figure out what the codons are. You have to figure out how this is divided into words. So we separate the sequence of letters into sets of 3, and we can see that the mutation here is in this codon. It changed the codon from CAG to CAC. Now we can look at the amino acid table, and we can check for these two codons, CAG and CAC, and we can see that the codon CAG is right here. That encodes the amino acid glutamine, whereas the codon CAC is, where are we? Oh, of course, right here. And that encodes the amino acid histidine. And so when you change that one letter out from being a G to being a C, the effect is that instead of the protein reading lysine, leucine, histidine, glutamine, serine, now it reads lysine, leucine, histidine, histidine, serine. This type of mutation is called a missense mutation. A missense mutation is a swapping out of one letter in the DNA, which leads to one amino acid being substituted for a separate amino acid. In this case, a glutamine amino acid was changed to a histidine amino acid. So in this checkpoint, what I want you to do is look at the codon table, which I'll provide on the next slide, and give me an example of a mutation that would be considered a missense mutation, any example. How I want you to provide this answer is give me one codon here, and then show me what it would change to in order to create a missense mutation. And remember, you can only swap out one letter in that codon. So that's one type of mutation. Let's look at another one. Here we have, again, the original and the mutant. This time I've changed out a U for a C. The way to approach this question is, again, to divide the sequences up into sets of 3-letter words. And now we can see that the mutation is affecting this codon right here. It is a UAC instead of a UAU. We go to the codon table. We see that UAC encodes for tyrosine, whereas UAU also encodes for tyrosine, right? And so this mutation right here, although we changed out a letter, it actually had no effect on the protein that was made because both UAU and UAC encode the amino acid tyrosine. This type of mutation is called a silent mutation. Silent mutations are an artifact of the reality that multiple codons end up translating into the same amino acid. Because of this, some changes in one letter for the other in the DNA actually have no effect on the amino acid sequence or the protein. And these mutations are therefore called silent mutations because although they are there, they go undetected. Just like in the previous checkpoint, here I would like you to give me an example of a codon changing into another codon and in a way that creates a silent mutation. So one codon here, change out one of the letters for the next codon, but it can't change the amino acid. Here's one more type of mutation. So here we have the original. In the mutant, I've swapped out a C for an A. We divide it up into sets of 3 letters, and now we see that we've turned a UGC into a UGA. Whereas UGC encodes the amino acid cysteine, UGA is actually a stop codon. And so what this means is that the sequence of amino acids that will be translated in the original will be phenylalanine, cysteine, lysine, histidine, proline, but in the mutant, it'll go phenylalanine followed immediately by a stop. And the other three codons will be ignored because the ribosome doesn't know that this is a mistaken stop codon. It just sees the stop codon and it stops translating and goes to termination. These types of mutations are called nonsense mutations. A nonsense mutation turns a normal codon into a stop codon. And these types of mutations can be particularly bad because they cause an entire chunk of the protein not to be made. They are especially bad when they occur towards the beginning of the protein because they can cause even the majority of the protein to not be made, which can have a very dramatic effect on cells. Once again, in the style of the other two checkpoints, using the codon table, tell me an example of a mutation that would be considered a nonsense mutation. Now we have one other type of mutation to talk about here. I've got the original sequence up here and the mutant down here. And in this case, instead of swapping out a letter, I actually added a letter in. So the original goes ACU, AUA. Down here, I've got ACU, AGUA. So this sequence is one letter longer than this one. What effect will this have on the amino acid sequence of the protein? Well, first we have to divide this up into three-letter codons. And now we see that the effect of inserting that one letter does not just change the codon where it was inserted, which turns from AUA to AUGU, but rather the effect of inserting it affects all of the codons that are after it and makes them no longer match the original two. That means that when the amino acids are translated, that every single one of them after this point is going to be something different. Only the first amino acid, the one that came before the mutation, is unaffected by the insertion of this extra letter. These types of mutations are called frameshift mutations. Frameshift mutations are what you get when one or more nucleotides is inserted or deleted. We saw an insertion, but this effect also happens if you delete letters as well. The effect is that the reading frame of the DNA codons is shifted, which is why it's called a frameshift mutation. This impacts the translation of all of the downstream codons. You can imagine how this works if you take an example in the human language. I've got a sort of nonsensical sentence here made of three-letter words. If I take these letters here and I split them up into three-letter segments, we get the dog ran for his log, and that is sort of a sensical sentence. But what happens if I take another letter and I insert it right at the beginning? Well, then if I ask you to slice this sentence up into three-letter words, now we have something that is totally different and makes no sense. So this is the basis for frameshift mutations, which can also be very dramatic and have very negative consequences for cells. So in the last part of this lecture, in order to really reinforce these concepts of mutations, we are going to go through two different sort of case studies of mutations, one of which is a mutation that is inherited, and another one is a mutation that is acquired. An inherited mutation is a mutation that is passed down from an organism's parents. An inherited mutation affects the DNA found in all cells in your body. And the reason why is because that mutation was carried in either the sperm or the egg, which fertilized in order to become the zygote, and then that zygote was the basis for all of the other cells in your body which were copied from it. So the mutation gets propagated through every cell in your body if you have it from the moment of conception. Acquired mutations, on the other hand, are mutations that can arise in individual cells in your body and then spread as those cells multiply. These mutations can come from either random errors that your cell makes or exposure to mutagenic substances, which can increase the rate at which your DNA mutates. And we'll talk about what some of those substances are later on in the lecture. So first we're going to talk about an example of an inherited mutation, and then we'll talk about an example of an acquired mutation. The inherited mutation that we'll talk about is the mutation that causes the disease sickle cell anemia, which we've referenced several times before. Sickle cell anemia is caused by a mutation in the protein hemoglobin, which is responsible for the very important job of carrying oxygen through your circulatory system from your lungs to all of the body's tissues. Every red blood cell carries hundreds of thousands of these hemoglobin proteins, so they make up a very significant part of the interior of the protein. Now, here's what the genetic sequence looks like for a normal hemoglobin protein. Here are the letters that make it up. People who have sickle cell disease inherit 2 copies of this sequence that, instead of looking like this, look like this. And if you didn't see a difference, I'll highlight it for you. One letter is different up here. Instead of being an A, it's a T. Now, I've taken these letters and I have translated them, or pardon me, transcribed them for you here into a messenger RNA sequence. This is the sequence of the first 7 codons of the allele for normal, healthy hemoglobin protein. And down here, we have the sequence for the abnormal hemoglobin protein. So what I want you to do in this checkpoint is translate for me both the sequence of amino acids for the normal, healthy hemoglobin protein and the sequence of amino acids for the abnormal, diseased hemoglobin protein. So write down these 2 sequences, divide them up into sets of 3 letters, and then use the codon table to come up with the sequence. So although I won't tell you where it happens, somewhere in your amino acids that you wrote down for that last checkpoint, you should have seen a change in one amino acid from being glutamic acid, which you can see highlighted here at the bottom, to being a valine, which you can see highlighted here at the top. You will note that these 2 amino acids occupy different sort of chemical categories on the amino acid table. Glutamic acid down here is electrically charged, whereas valine belongs to the nonpolar category and is therefore hydrophobic, meaning that it doesn't like to get together with water. Electrically charged things have no problem with water, and so the effect of changing the glutamic acid to a valine actually completely changes the way that the protein is structured. A normal hemoglobin protein looks like this, and it has multiple versions of this protein that are stuck together, and they have their glutamic acids facing outward towards the watery cellular environment, which is not a problem when that amino acid is glutamic acid because electrically charged amino acids don't have a problem with water. However, you can imagine what happens when this glutamic acid is changed into the amino acid valine. Valine is going to act like oil, another hydrophobic substance, and try to separate itself out from water. This changes the way that the protein folds, and it actually causes multiple hemoglobin proteins to attract each other and stick together so that they can shield their valines from exposure to water. The result is that while normal hemoglobin proteins are freestanding like this, sickle cell causes the hemoglobin proteins to clump in this manner here because of their one hydrophobic out-of-place amino acid. And this clumping activity is what causes the overall shape of the red blood cell to form the sickle. Of course, this sickle shape causes problems for blood flow because it can block arteries and veins, And this is what can lead to all of the various symptoms associated with sickle cell disease, these abnormally shaped red blood cells, which trace their shape back to the activity of the protein, which is caused by the mutation from that one amino acid to a different hydrophobic amino acid. So, knowing what you just learned about the basis for sickle cell disease, in this checkpoint, I want you to use the codon table and the amino acid chart to figure out which of the following mutations would have a greater, or in other words, a worse impact on an organism. And why would that be the case? So the first step will be to write these mutations down. Then I want you to use the codon table to figure out what the mutations change the amino acids into. And then finally, look at what categories those amino acids belong to and figure out which one would have a more dramatic effect and why. So that completes our discussion of our case study on an inherited mutation. And now we're going to talk about an acquired mutation. Because as we know, while mutations can be passed down from your parents, they can also arise in individual cells as a result of mistakes that are made during DNA replication, during S phase of the cell cycle, or exposure to environmental mutagens. When these mutations specifically affect a gene that is important for regulating the cell cycle or suppressing tumors, the result is cancer, as we've talked about before. So in this very last part of the lecture, we are going to walk through a real-life case study of a woman who had a specific type of cancer that was related to her RB1 or her retinoblastoma protein tumor suppressor. Now, it used to be far less common for patients to have an actual understanding of the basis of the genetics of their cancer. But it is becoming more and more common for doctors to actually look at the DNA of the cells in a tumor and figure out what mutations are causing the cancerous cells to arise. In this case, we have one such study of a woman named Mary, who was a 42-year-old mother of 3. It's not her real name. And she had smoked cigarettes since she was 19. But what was concerning for her and what brought her to the doctor was that she developed a persistent cough some time ago, and in the last few days she had developed tremors in her hands and other neurological symptoms. In this case study, they explained that she went to the doctor, and although her lung x-ray from 2 years prior had been clear, her x-ray upon this visit showed a tumor, which can be seen in the left-hand side of this picture. At this point, she was given a biopsy, and the biopsy revealed a particular type of cancer called squamous cell carcinoma, which can be seen here and here, and you can see the rapid growth of the cells and the concentration of those cells because they are dividing so quickly without having anything to regulate the progress through the cell cycle. They also found that the cancer had metastasized, which means that it had moved through the bloodstream or the lymph nodes and had been carried to another part of the body. And in this case, the cancerous cells had moved into the brain, where there was a metastatic tumor located which was causing the neurological symptoms and the hand tremors. Now, in Mary's case, her cancer was caused specifically by a mutation in the gene for retinoblastoma 1, a tumor suppressor protein that we learned is involved in the G1 checkpoint of the cell cycle, specifically in checking for adequate cell size before proceeding into the next stage. Now, the retinoblastoma protein instructions are normally almost 5,000 letters long. There's a piece of DNA in all of our genomes that's 5,000 letters long, and it encodes instructions for making this special protein which regulates the cell cycle. I have shown you here only a segment of those 5,000 letters, and as you can see, we make it up to about letter 2600, meaning that we're approximately halfway through the protein when Mary's mutation arises. I'll go back and forth between the last slide and this one again. You can see that normally there's a G there, But when Mary's cancerous cells were sequenced, they found that that G was no longer there. So in this checkpoint here, I want you to tell me, how would her mutation be classified? What type of mutation is it? And what would be the effect of this mutation on the RB1 protein that her cells are making? So obviously, this mutation caused a non-functional version of the protein to be made and allowed the cell cycle to get out of control, resulting in the cancer. But what caused that mutation to arise in the first place? Well, the overwhelming likelihood is that various compounds inside of the cigarette damaged her DNA at the location of this particular letter in the RB1 gene. Substances that mutate DNA are referred to collectively as mutagens, and substances that mutate DNA in ways that cause cancer are called carcinogens. Here are some of the more well-known, well-understood mutagens and carcinogens. We, of course, have cigarettes, we have alcohol, and we also have, this one you may not know about, cured meats. Cured meats are meats that are preserved through the addition of salt. So this includes things like bacon, pancetta, sausages, and it was discovered that there is a correlation between consumption of cured meats and cancers of the stomach, colon, and rectum. So that is a significant contribution. And of course, Arizona's most famous mutagen, down here in the desert, is the sun. UV rays are capable of damaging DNA and causing mutations because what UV energy is actually capable of doing is when there are two T's adjacent to each other in a sequence of DNA, then the UV radiation causes those two T's to fuse and into a structure that is called a thymidine dimer. And of course, then when the DNA replication enzymes come along to make more DNA, they get confused by this structure and they make all sorts of errors, which lead to mutations. And I'm here to deliver some bad news to you, which is that there is really no safe way to get tan skin because it is DNA damage itself that causes tanning to happen. Tanning is a reaction to damage that has been done to your DNA. And so by the time you're getting tan, regardless of what method, regardless of whether it's in a tanning bed or in a salon or just outside by the pool, regardless of how you tan, if you're getting tan, it means that damage has been done. So on that sort of not so cheerful note, we will finish the lecture, and I will see you in the next one.